All optical engineers start by understanding first-order or paraxial optics. This simple view of optics allows for us to find where the ideal object-image relationships are and quickly estimate where rays from a source will end up. But this picture quickly becomes blurry, pun intended, when we consider the path that all rays take from an object. In fact, due to a variety of errors called aberrations, not all of the light from a singular point is imaged to another singular point. There are a variety of aberrations that result in imaging errors, but chiefly, the third-order aberrations, known as Seidel aberrations, can be understood as how the path of rays differ from the ideal. The specific aberration can be determined by the normalized pupil coordinate of the ray, which is expressed in Cartesian coordinates xp and yp, or in circular coordinates rho and theta, and also the normalized image height h. We can think of aberrations as wavefront errors or ray errors. Wavefront errors are thought of as the optical path difference between an actual wavefront exiting the exit pupil and the ideal reference wavefront, which would focus perfectly on the reference paraxial image point. Meanwhile, ray errors are displacement from the ideal ray imaging point and are noted as epsilon x and epsilon y, respectively, in the transverse plane. Meanwhile, if a ray crosses the optical axis at a different location relative to the paraxial equivalent, it is said to have longitudinal aberration, which is denoted as epsilon z. Because many optical systems are rotationally symmetric, ray fan plots of transverse optical aberration provide essential information as to how light across a pupil dimension will be aberrated. Specific aberrations will depend on pupil location and image height. For example, a common first-order aberration is defocus, which is simply displacing the image plane from the ideal image plane. Meanwhile, a common third-order aberration is spherical aberration. Spherical aberration results from rays at the margins of the pupil plane being imaged before the chief ray. In fact, the marginal ray crosses the optical axis before the chief ray. The locations they intersect the optical axis are called the marginal focus and paraxial focus, respectively. Based on the predicted equation derived from the wavefront error, we can calculate the spherical aberration for an optical system. As shown above, we see that spherical ray aberration for a system has a cubic dependence on pupil coordinates. Interestingly, ray aberrations add linearly to one another. For example, adding defocus, which has a linear dependence on pupil height, as well as the spherical aberration, has a ray fan plot that adds to this. The same can be done for tilt, which has a transverse ray aberration that are only linearly dependent on image height. By splitting the power as much as possible among lens surfaces, we can reduce aberrations like spherical aberration. This equation shows the total wavefront spherical aberration that occurs from a lens. By understanding the conjugate factor, which is the change in slope of an incoming and outgoing ray from a lens, we can pick the shape factor b to minimize the wavefront error. For example, a conjugate factor of -1, which takes a collimated beam and focuses it, the ideal shape factor for a conjugate factor of -1 is 0.741, meaning the lens is nearly convex plano. Using this theory, we got into the lab. All right, our 90-degree turn has been achieved with our pentaprism. We first align our laser along this rail, and using our pentaprism and ensuring that the beam goes through the center of the pinhole aperture at the beginning and all the way at the end.
So we have aligned our pentaprism to our mask, and the mask also to the lens. So all of our system is aligned, as you can see there. And we now decided our numerical aperture. How did we decide that?
We decided it by checking the numerical aperture of the lens and ensuring that our objective lens has a larger numerical aperture to make sure we're not cutting off any light that could be going into the system.
So what's our numerical aperture we're using?
0.4 is for the microscope objective. That's with a 0.33 numerical aperture test lens. What is up, guys? We are going to find the marginal focus next. We have an aligned system. As you can see very faintly, we have our crosshair almost directly, or actually directly on top of where our beam was, along with the spot. And now we're going to find our marginal focus by going to the very far edge of the Shack Hartman on the lens and moving the objective until we get to the marginal focus. So we just determined that the far left of the Shack Hartman will be our marginal ray. The objective was slightly in front and focused slightly in front with this ray as compared to the far right of the Shack Hartman. So this will be our starting point for our marginal focus.
So now for this part of the lab, we scanned our pentaprism through each one of the aperture holes in the mask and adjusted our microscope location laterally to or perpendicular to the rail location, so that each time the spot was in the center, we recorded the microscope location and the location that we adjusted our pentaprism. So for this part of the lab, we turned over our lens so that the more curved side is facing our pentaprism, and then we did the same type of steps that we did previously in lab part B, where we moved the microscope perpendicular to our optical rail to then determine our steps on our different modes, and using that we can see how lens shape factors affect spherical aberration.
Hello everyone, we are translating our microscope along the optical axis to find our longitudinal aberration, which is defined as the distance between our paraxial focus and our marginal focus. So these two aberrations are where the marginal ray crosses the optical axis and the chief ray crosses the optical axis.
Now we will move on to our results from this lab. First, we applied the normalized hole positions to the normalized microscope positions. To normalize the hole positions, we labeled the two extreme hole positions -1 to 1, since there were 9 positions and the increments were in steps of 0.25. We normalized the microscope positions by subtracting the middle hole microscope position to all of our values. This gave us a ray fan plot for the transverse and sagittal ray aberrations. They are the same plot because of the cubed nature of spherical aberration. Using Zmax, we can compare our plots to a theoretical version. As you can see, our measured ray fan plot does have the same shape. However, some of the values are slightly off. This could be due to a variety of reasons involving human errors such as reading the measurements incorrectly or mathematical errors in processing. Beam alignment could have also been a source of error. This first measurement occurred when the planar side of the lens was towards the pentaprism or the source. This caused us to have a shape factor of -1, which we found in this equation, where r is the radius of curvature. You can also see it when looking at this image. Upon adjusting the lens to achieve a shape factor of 1, we observed our normalized values in relation to our theoretical expectations. Similar patterns emerge, yet discrepancies exist, likely due to the previous mentioned errors. Comparing the shape factor plots reveals reduced spherical aberration at a shape factor of 1 compared to -1, illustrating the lens light bending characteristics. Optimal spherical aberration minimization is achieved when light rays bend equally at both surfaces. With an object positioned at infinity and a refractive index of 1.5, the ideal lens shape to employ is close to a convex plano lens, which should minimize spherical aberration we have determined. Next, we examined the longitudinal aberration normalized plots for the plano-convex lens and compare them to the theoretical predictions. The observed discrepancies in shape may stem from alignment issues. Notably, imaging the paraxial ray represented by the center hole in the Shack-Hartman mask proved challenging. In our first analysis, we utilized the displacement data from the marginal ray to the paraxial ray to estimate the system's defocus, calculating a defocus of 12.4 micrometers. This measurement indicates the displacement of the actual image plane from the paraxial image plane. Understanding defocus is crucial as it enables the adjustment of the image plane or reference image point, facilitating aberration correction and enhancing image quality.